Hello everybody, I'm Dr. Mohammed Waqas. Today we are going to talk about erythropoiesis. In previous lecture we discussed site of erythropoiesis, and in this lecture we have to discuss process of erythropoiesis, how the formation of erythrocyte take place in our body. So students, previously we discussed bone marrow is the main site for the formation of all type of blood cells. So students, these hematopoietic stem cells present in the bone marrow. These are the primitive cells which give rise all type of blood cells in the bone marrow. These hematopoietic stem cells in the bone marrow also called uncommitted pluripotent hematopoietic stem cells. These cells also give rise all type of blood cells, but in early stage they are not designed to form all type of blood cells. Students, the main source, the best source of this cell is umbilical cord of blood. So students, when they are designed to form all type of blood cells, these uncommitted pluripotent hematopoietic stem cells change into committed pluripotent hematopoietic stem cells. So there are two types of these cells. First is lymphoid stem cells, and second is colony forming blastocytes. These lymphoid stem cells form lymphocytes and natural killing cell. And the second is colony forming blastocytes. These colony forming blastocytes form myeloid cells. These are the blood cells other than lymphocytes. So when these grown in culture, they form colonies. There are different unit of colony forming cells. First is colony forming unit of erythrocytes. Second is colony forming unit of granulocytes. And on the third is colony forming unit of megakaryocytes. So students, these colony forming unit of erythrocyte develop into erythrocytes. And on the second is colony forming unit of granulocytes. These are developed into neutrophils, basophils, and eosinophils. So on the third is colony forming unit of megakaryocytes. Friends, these colony forming unit of megakaryocytes develop into platelets in the bone marrow. So after that we have to discuss different stages of erythropoiesis. Now we have to discuss stages of erythropoiesis. Students, the formation of erythrocyte take place in 6 stages. First is pro-erythroblast, also called megablast. On the second is early normoblast. After that is intermediate normoblast. On the fourth number is late normoblast. And on the fifth is reticulocytes. And at the end is mature erythrocytes. Students, in pro-erythroblast, cell derived from colony forming unit of erythrocytes. So they are very large in size. The diameter of this cell is 20 micro. Students, the nucleus of these cells is very large. So there are two or more nuclei and reticular network is present. Since in this stage, synthesis of hemoglobin start. Synthesis of hemoglobin start in this stage. So the cytoplasm, because there is no hemoglobin is present, that's why the cytoplasm, the pro-erythroblast is basophilic in nature. After that is early normoblast. Students, this is little smaller than pro-erythroblast. So that diameter of this cell is 15 micro. And the nuclei disappear in this stage. Nuclei disappear in this stage. We discussed there are two or more nuclei and reticular network are present, but in this stage nuclei disappear. So condensation of chromatin network start in this stage. So cytoplasm is basophilic in nature also in this stage. And after that is intermediate normoblast. So it is also smaller than early normoblast. The diameter is 10 to 12 micro. Hence, the condensation of chromatin network continue in this stage, and also in this stage hemoglobin appearing start. So because in this stage, pairing of hemoglobin starts, so that's why cytoplasm is also acid and basic in nature. Because it is already basophilic, that's why, because in this stage hemoglobin pairing starts, so this is acid and also basic in nature. On the fourth number is late normoblast. Friends, in this stage, also the diameter of the cell decrease and become 8 to 10 micro. So there is very much condensed chromatin network. So, and also a large quantity of hemoglobin present in this stage in late normoblast. A large quantity of hemoglobin present. So that's why the cytoplasm in this stage is acidophilic in nature. So after that is reticulocytes and mature erythrocytes. So after that is reticulocytes. In this stage, cells are also called immature erythrocytes. These are the slightly larger than mature erythrocytes. Students, because the cytoplasm contain reticular network and reticulum, that's why we said that reticulocytes. Students, in newborn baby, 2.6 RBCs are present in reticulocytes. Students, due to presence of remnants, disintegrated Golgi apparatus, and other organelles, the cytoplasm of reticulocytes is basophilic in nature. So on the sixth is matured erythrocytes. Students, in this stage, reticular network disappear and the cell become mature in this stage. Students, they attain biconcave shape and the diameter of matured erythrocytes is 7.2 micro. Students, from up to reticulocytes, he needs 7 days. Sorry, 5 days. And to become mature, he need 2 days. So totally, in this cycle, he needs 7 days.